To differentiate between areas that are sources of attention, which drive attentional modulations elsewhere in the brain, and areas that merely receive these signals, you need to do more than just record neural activity. You need to control neural activity, to causally intervene, either driving or suppressing activity in a particular brain area, and then examining the effect of that manipulation on sensory signals or attentional modulation in other areas. Let's talk about a couple methods for carrying out these types of causal manipulations and what they've showed us about the areas that serve as sources of sensory modulation during attention. Electrical stimulation, via an electrode inserted into the brain, can be used to directly induce neural activity. This technique has been around since the 1800s and was one of the first sources of evidence linking particular parts of the brain to specific functions. Recently, electrical stimulation has been used to test the relationship between neural activity in one brain area and processing of visual signals in another part of the brain. So, to see whether inducing activity in one brain region is sufficient to recreate the changes seen during an attention task. There are also some therapeutic uses for this technique in humans. For example, deep brain stimulation is used to relieve the symptoms of Parkinson's disease. As discussed in the previous video, some areas within prefrontal cortex signal the location of attention. One of these areas is the frontal eye field, or FEF. Because the FEF is retinotopically organized, just like the visual responses that we discussed before, electrical stimulation can be targeted to just one part of space. Electrical stimulation of the FEF has been shown to improve thresholds for change detection, similar to a cue in an attention task. And this improvement is specifically at the part of space represented at the stimulated location in FEF. The stimulation also increases the strength of visual signals at the corresponding location in other brain areas. It's also possible to directly infuse small volumes of drugs into the brain tissue in order to manipulate neural activity. These tiny, localized injections give you more specific information about the site of drug action in the brain than you would get administering the same drug orally or injecting it into the bloodstream, when it could affect neurons spread throughout many different brain areas. Drugs can bind to neurotransmitter receptors and either mimic or inhibit the normal signaling through that receptor, which will either cause or prevent neural activity near the injection site. Local injections of drugs that inhibit neural activity into prefrontal cortex have been shown to disrupt performance on attention tasks, and also to reduce the strength of responses in visual areas, consistent with the idea that these prefrontal areas are the source, which converts an attentional cue into modulations of responses of incoming visual stimuli. There are a few other options for activating or silencing neurons. For example, you can get the neurons to produce an ion channel that opens in response to light, and then control their activity with light. Cooling an area of brain tissue will temporarily silence those neurons as well. And you can lesion neurons, either with a comparatively crude physical removal of brain tissue or with a genetically targeted ablation of those cells. While most of these techniques can only be used in animal subjects, there is at least one technique that can be used to manipulate neural activity in healthy human volunteers and observe the effect on behavior and activity in other regions of the brain. This is called transcranial magnetic stimulation, and it's a non-invasive method that uses an external coil to generate a changing magnetic field, which will then induce electrical currents in brain tissue. One study showed that TMS targeted at a particular area of the prefrontal cortex in humans increased the perceived contrast of stimuli, again, reminding us of the effects of a spatial cue. The stimulation also increased visual responses to visual stimuli as measured with simultaneous fMRI of visual areas of the brain. In sum, these causal experiments indicate that the prefrontal cortex modulates sensory activity elsewhere in the brain during selective attention.